ASRB Net Plant Pathology Preparation Model Question Paper 19. I'm in Shankar Reddy, PhD Plant Pathology. First one is which of the following is a seed-borne rust? Coffee rust, sunflower rust, safflower rust, linseed rust. Coffee rust is Hemilia vastatrix, that is airborne. Sunflower rust, linseed rust also. But when among the rust, this safflower rust is actually seed-borne and also spread through other means. But the answer for the option is question is C, safflower rust. Second one is a fusarium is a seed-borne, soil-borne, airborne, waterborne. We know very well fusarium is a strictly soil-borne, that it produces three kinds of spores: microconidia, macroconidia, and chlamydospores. Right. The next one is a bimodal curve is the characteristic feature of monocyclic diseases, polycyclic diseases, polyetic diseases, all the above. So to understand this, I gave a small table here. Monocyclic diseases, polycyclic, polyetic. Monocyclic diseases, the rate of inoculum is 0.02 units per day, whereas polycyclic diseases 0.1 to 0.5 units per day. When it comes to polyetic diseases, the rate of inoculum is 0.02 to 2.3 units per year, not day. So monocyclic disease produces a saturation curve. Polycyclic diseases produces a sigmoid curve or S-shaped curve. Polyetic diseases produces a bimodal curve. So these are all the examples. What they asked is a bimodal curve is produced by. Bimodal curve is actually produced by polyetic diseases. And examples also given here. Monocyclic disease examples is smut, root rot, wilt kind of diseases. When it comes to polycyclic diseases, powdery mildew, rust, leaf spots, it is especially airborne disease, and late blight of potato, rice blast are the example of polyetic disease. When it comes to polycyclic disease, when it comes to polyetic diseases, CTV, Cetusticida virus, Dutch elm disease, fungal vascular wilts, and mycoplasmal yellosis are example for polyetic diseases. The next one is the use of agar as a solidifying agent in culture media was suggested by Fra Hesse, Walter Hesse, Robert Koch, and Robert Petrie. So the use of agar in the culture media was first suggested by Fra Hesse. She is the wife of Walter Hesse. This Walter Hesse is an assistant of Robert Koch. This Robert Petrie is an assistant of Robert Koch. I will tell you. So actually this Fra Hesse has a little bit experience in this microbiology, especially in laboratory related things. So while she was working with various other kinds of things, especially home making some kind of things, so she discovered that this agar having solidifying property. He immediately told to her husband Walter Hesse. So he just again started working on it and finally came to conclusion that the agar can act as a solidifying agent, and told this to Robert Koch, where this Walter Hesse is working in the laboratory of Robert Koch. This Robert Petrie actually who discovered the Petri plate is also the assistant of Robert Koch only. So before inventing the agar, gelatin is the agent the researcher used to use for the solidification. But gelatin was actually liquefied and digested by some kind of bacteria, especially at high temperatures. But there is no problem with this agar because agar can withstand up to 98 degrees Celsius and can only melt at 42 degrees Celsius. So to overcome the problem, the gelatin was replaced with this agar, which is a solidifying agent. It was first discovered by Fra Hesse. If there is no option of Fra Hesse, then you can go with Walter Hesse. The next one, TMV cryptogram. So to understand what is cryptogram, I have given something here. So cryptogram is generally a short form of information about virus properties by giving various kinds of symbols, and all those things will be there. So to understand this, what is this R1, 2, 5, something like that will be there, right? So the first pair of information, R stands for RNA, D stands for DNA, and the number of strands in nucleic acid, single stranded or double stranded, something like that. If we can see here, R stands for RNA, 1 stands for 1 nucleic acid. So second pair, molecular weight of the nucleic acid in a million Daltons. The second one is the percentage of nucleic acid in infective particle. So 2 is the molecular weight, 2 million Dalton. 5. So 5 is the 5% is the nucleic acid percentage of the virus. For example, 20% nucleic acid is there in the virus, 20 or 10% means 10, 5 means 5, like that. So TMV generally contains 5% nucleic acid and 95% protein code. The next one is the third pair, the outline of the virus particles under the nucleocapsid. S means elongated, E means S means spherical, E means elongated or rod like, U means again elongated, X means complex, B means bacilliform. So here E means elongated, again E means elongated. The last one is the fourth pair. It's a kind or host infected nature of a virus transmission or a kind of vector. So what kind of vector it is involved? For example, S means a sap transmission, like that. Host means O. The last one is again host. So something like that. They used to give in a short form, that is called cryptograms, by denoting their properties, giving the symbols. Generally the TMV cryptogram is R125ESO. This is the general cryptogram of TMV. Don't worry about it. Nowadays we are not using any cryptograms anymore. The next one is thread-like viruses are potiviruses, calimoviruses, clustroviruses, bimoviruses. So generally these clustroviruses are actually look like a thread or a tapeworm like viruses. These are all clustroviruses, and it's also the longest, And who was the first to recognize and delimit the ascomycetes as a distinct group of fungi? August Carda, E.J. Butler, Linnaeus, Leeuwenhoek. Actually, this was first observed by August Carda. So, where he delimited the ascomycete as a distinct group of fungi. Now we know that ascomycete is one of the largest group of fungi, which contains thousands of fungi, right? This one is a motile haploid zoospore. I repeat, motile haploid zoospore. Now we'll see zoospores are haploid, motile. Aplanospores are, they are haploid but non-motile. Aplano means non-motile. Planospores are motile, right? Conidia is haploid but non-motile. Zygospore is diploid and sexual spore. It is what they asked is a motile haploid spore. So among the given options, zoospore is the best example for motile haploid spore. Of course, other spores are like aplanospore and conidiospores are also haploid, but they they don't have any motility structures like flagella and all those things, so they cannot motile. So the answer is oospore. Zygospore is a diploid resting spore. So to eradicate black rust, Barbary eradication campaign was done in USA during the year around 1918. They have done a Barbary eradication program, but this USDA, United States Department of Agriculture, started a Barbary eradication program in US around the 1970s, and they started around 2 to 3 million trees are eradicated, what they have mentioned in the APS website. But this eradication program, the original campaign was started in 1918. Which of the following citrus species restrict the entry of Xanthomonas citri through stomata? Which means, so generally this pathogen enters through stomata. So which species of citrus can restrict this stomatal entry? Lime, mandarin, sweet orange, trifoliate orange. Generally, this mandarin species actually restrict the entry of this Xanthomonas citrus canker species through stomata. So the next one is the germinating teleospore is known as hypobasidium. You guys just also go through the other terminology like what is epibasidium and what is probasidium. There are very few terminologies only. So the answer is hypobasidium, because we have a very less time. I can't able to explain every single thing. And the next one is which of the following is not the student of Anton de Berry? H.M. Ward, A. Fisher, MS Voronin, and E.F. Smith. H.M. Ward is a student of Anton de Berry. Voronin also a student of Anton de Berry. Fisher is also a student of Anton de Berry. So the answer is E.F. Smith. He is not a student of Anton de Berry. The basidospores are violently discharged in a wind, water drop mechanism, both A and B, none of the above. In water droplet mechanism, the basidiospores are violently discharged because that water pressure that will actually burst like with force, so that with the water droplet the spores also can be released. Wind also may be the example of violently discharged. Probably A and B. I'm not sure of this question. Probably A and B, both wind and water droplet mechanism where the basidiospores are discharged violently. The next one is budness fungi, stink horns, puffballs. Coral fungi are the members of Ascomycota, Basidiomycota, Zygomycota, Oomycota. Majority of the mushrooms are comes under Basidiomycota, but few mushrooms also comes under Ascomycota also. But this budness fungi, stink horns, and all the other fungi actually comes under Basidiomycota group of fungi. Probably Basidiomycota group of fungi. As of course, maybe Basidiomycota only. Big bed of tomato is caused by Phytoplasma, virus, bacteria, fungus. Big bed of tomato is caused by Phytoplasmal pathogen. And for our understanding, I have given some other things like confusing things when it comes to bunch top. Tomato bunch top is caused by viroid, whereas banana bunch top is caused by virus. Papaya bunch top is caused by FEB, Lactoferous FEB, fastidious vascular bacteria. So these are little variations when it comes to bunch tops. The term heterokaryosis was used in the case of Beta infestans, Rhizophus, Rhizoctonia solani, botrytis cinerea. It is related actually botrytis cinerea because this term heterokaryosis and the phenomenon of heterokaryosis was discovered Hanston Smith in 1923. Probably 1932, not sure. In botrytis cinerea only, but the answer is botrytis cinerea, that I am sure. Next one is coconut stem bleeding is caused by Phytophthora parvivora, Ceratocystis paradoxa, Phytophthora cactorum, all the above. So coconut stem bleeding is caused by Ceratocystis paradoxa. Witch's broom of cocoa is caused by, generally all witch's brooms are caused by fungi only. Generally you can take on the right side, I have given peach leaf curl is caused by Taphrina deformans. Witch's broom of cherries is caused by Taphrina epiphylla. Turmeric leaf blotch is caused by Taphrina maculans. There are some Taphrina diseases. Who here generally witch's broom of cherries is caused by Taphrina epiphylla, and witch's broom of cocoa is a fungal pathogen. Which of the following organism commonly affects tomato, cotton and banana? Fusarium moniliforme, no, Fusarium oxysporum, Alternaria solani, Erythropholigon. Yes, it is related to Fusarium solani. It causes tomato anthracnose, cotton, banana. So all these are related to that can cause the diseases in these three crops. Next one is fungi are unicellular, multicellular, acellular, and all the above. You know very well that fungi are the best example for multicellular organisms. The crop varieties are resistant to wilt due to the presence of HCl nitrogen cyanide in root exudates. So cotton, sunflower, soybean, linseed. So answer is actually linseed because the linseed roots contains hydrogen cyanide, so that in linseed the wilt causing the wilt disease is very rare, and most of the cases we cannot see wilt because the root exudates contains hydrogen cyanide. It can easily inhibits or kills the wilt pathogen. Next one is bacterial brown rot and potato wilt pathogens produce predominantly severe symptoms due to the production of the following compounds. So what they are telling is brown rot pathogen and potato wilt pathogen produces very severe symptoms due to the production of which of the following thing. So the following compounds: proteins, lipids, gibberellins and polysaccharides. Generally this bacterial brown rot pathogens and potato wilt pathogens produces polysaccharides that can cause very severe damages or very severe symptoms. So the answer is polysaccharides. Which of the following method is suitable for the production of virus free plants? Tissue culture, pollen culture, embryo culture, meristem tip culture. Tissue culture can be recommended, but for the production of virus free plants the most recommended method is meristem tip culture, because the meristem is actually divides continuously. It's a continuously dividing tissue where the virus cannot stay. So the best method is meristem tip culture. The next one is phylacteria causes powdery mildews, downy mildews, leaf spot, leaf blights. Generally phylacteria causes powdery mildews because there are some genera For example, if you take downy mildew genera, Peranospora, Sclerospora, Paranosclerospora, Bremia, like that, some kind of genera is there, generally causes downy mildews. When it comes to phylactinia causes powdery mildews. For example, Ericife, Levelula, Phylactinia, Podospora, Sparotheca. These are all the few genera which generally causes powdery mildew. So this Phylactinia is a genus of generally powdery mildew. Then 1 gram in fungicide in 1 liter of water equal to probably 10 ppm. So it will come 1 gram fungicide in 1000 ml of water. 1 liter of water is known as, generally it comes to 1000 ppm. It which region of genome the pathogen preferred for the variation and determination. So generally TDNA region, ITS region, CCR region, LRR region. So for fungal variation detection, or for the detection of fungal variation, we can recommend ITS, internal transcribed spacer. You can take animal is COI, KIT 12S, 16S. When it comes to plants, MATK, RBCL, ITS is generally mostly recommended for fungi. And when it comes to bacteria, cole, ROBP, 16S, CPN60, RIF. So for fungi, ITS, it is a very important. ITS, RPB1, RPB2 and 18S. And for protist, ITS, COI, RBCL, 18S and 28S. These are all the some kind of markers or locus that we target. But generally for fungus, we used to go with ITS. That is the most recommended and most doing outside. ITS. The aerial survey first used in the infection of, so which disease that was first the aerial survey was identified used, barley yellow dwarf and Helminthosporium varese bipolaris. Helminthosporium made is a corn leaf blight, and Pymetotrichum omnivorum. It is Pymetotrichum omnivorum because the aerial survey are this aerial surveying program was actually started in India in Kerala district on coconut diseases. It was actually started by Pishroth in 1970. That's why he is considered as the father of remote sensing in India. So he was the one who first used the remote sensing or this aerial survey with the coconut disease, that is Pymetotrichum omnivorum. The fruiting body of the fungal genus Ericife. Ericife is a powdery mildew. So the powdery mildew generally produces Cleistothecium. What is Cleistothecium? A completely closed ascocarp is known as Cleistothecium. It will be like this. Cleistothecium. What is Apothecium? Completely opened ascocarp. What is Perithecium? Partially opened ascocarp. It's like a flask, like a almost a flask shaped thing with a narrow or circular mouth or slit opening, that is known as ostiole. So the answer is Ericife generally produces Cleistothecium. We might have studied this appendages, right? Cleistothecium with that kind of appendages, Levelula, Ancinella, something will be there. These are all the powdery mildew genera, right? That Levelula, all those things are powdery mildew genera. So this powdery mildew generally produces sexual fruiting bodies known as Cleistothecium. Next one is P truffles, endogonals, mucorales, pezizales, agaricales. These P truffles are actually comes under this probably endogonals, but some other truffles also comes under maybe pezizales and other kinds of things. But generally, these P truffles are endogonals. And which of the following structure is not a part of the infection? Aprosorium, attachment organ; Haustorium, nutrient absorbing organ; penetration peg. From Haustoria, a small peg-like structure will be developed that actually make a hole and penetrate inside the leaf structure. That is a penetration peg. These three are the part of infection only. But trichomes are generally hair-like structure that we can observe on the plants. We can clearly observe on tomato. Some varieties of tomato we can see very big hair-like structure, that is trichomes. So is actually this trichome is not at all a part of this infection. The remaining three is a part of infection. So the answer is trichomes. The disease which creating restrictions in exporting the wheat disease: stem rust, kernel bunt of wheat, wheat blast, molia disease. Generally this kernel bunt of wheat is a very important disease when it comes to quarantine, because this disease we can also call as mission impossible. It's a very difficult to identify and as well as to control. That's why it is known as mission impossible diseases. So the answer is kernel bunt of wheat. Which of the following is not known as a spore factories? Not known as spore factories. It is not known. Okay. So Clistothecia, it contains abundant of spores. Apothecia, it also contains spores. Perithecia, it also contains spores. Pseudothecia, yes. Pseudothecia is not known as spore factories. Okay, the remaining all contains, all produces abundant or plenty of spores, but this pseudothecia produces no spores. I mean, it will produce, but it's not that much. Flask shaped fruiting body or flask shaped fruiting body is known as pycnidium, sporadium, acerole, cinema. Generally this pycnidium is also look like a flask shaped with a narrow opening or circular mouth is known as ostiole. So the answer is a pycnidium. So it is exactly resembles to perithecia. So answer is a pycnidium. The next one is the SAR, systemic acquired resistance stimulating fungicide. This is actually a kind of a defense that is actually present in plants. Systemic acquired resistance, induced systemic resistance. These are all the layers of defense in plants. So generally that can be stimulated by the application of fungicide, which means which fungicide can stimulates this is systemic acquired resistance in plants. Mancozeb, Benomil, S-benzol or S-methyl, all the above. Actually, it's S-benzol or S-methyl. In epidemiology also, I have given in my book that very clearly the difference between ICAR and ISR, where I have mentioned this S-benzol or S-methyl is a fungicide that is used to activate the SAR, systemic acquired resistance. The frequency of mutations in plant viruses. The frequency of mutations in plant viruses is very rare. It's just 0.1%. There is a chance of 0.1%. So most of the mutations in plant viruses are said to be as deleterious. Deleterious means harmful, not as for virus. Antibiotics has no effect on fungi, bacteria, phytoplasma, algae. Algae, even though you apply tons of antibiotics, there is no effect. So answer is algae. Complete or partial loss of virulence of a pathogen is known as attenuation. Attenuation is generally the term which is used for complete or partial loss of virulence. So again this virulence of the pathogen can be retained once we inoculate in the suitable host at a suitable environmental conditions. So generally this attenuation is a loss of virulence is known as attenuation. The term basidium was coined by Leville, Coda, E.M. Fries and Anton D. Barry. Generally, this the term basidium was coined by Leville in the maybe in the midst of 19th century. I'm not sure, but it was coined by Leville. The resting spores of potato what can survive in 10 to 20 years, 5 to 10 years, 20 to 35 years, but it is actually viable in for soil or maybe viable in soil for 10 to 20 years, but some of the books also mentioned like 5 to 10 years or 5 to 7 years, something like that. But you just take a look based on the options. But the standard one is 10 to 12. In Ascomycota or Ascomycetes, karyogamy occurred within Ascogonium, Antheridium, Ascogenous mother cell, Ascogenous hyphae. Actually, the karyogamy takes place in Ascogenous mother cell. So there are in if you see in free cell formation, that is ascus formation, otherwise called as free cell formation. In Ascomycota group of fungi, we are planning to publish a detailed video for each and everything. We are also planning to publish this ascus formation or free cell formation, where we explain in detail about how this karyogamy, where this karyogamy takes place, how the cells are forming and all those things. But when it comes to karyogamy, generally happens in a cell that is a ascogenous mother cell. This is the ascogenous mother cell. So from this ascogenous mother cell, small hair-like structure, this ascogenous hyphae, where this nucleus nucleus come in detached with, and there some other process will happens. But in this karyogamy generally takes place in this ascogenous mother cell. Virus medium can be obtained by filtration, heat radiation, autoclaving, heating. See, even though I only wrote the book, still I couldn't understand the meaning of this question, but the answer is given as filtration. I don't know what is this virus medium can be obtained through filtration and all those things. I was a little bit unclear about this. You guys please refer. Glasswares can be sterilized in oven at what temperature? This question was explained in the previous question paper number 18 also. I have given a list of hot air oven temperatures along with their timings. 80-20 hours, 120-20, 60, 162 hours, 182 hours. Generally, glasswares can be sterilized at a temperature of 160 degrees Celsius for 2 hours. The next one is virus is found in cells in the form of suspension form, liquid form, crystalline form, none of the above. Generally, virus is found in the cells in the form of crystalline form. Amphigenous type of anthracium is found in Pythium, Phytophthora, Plasmopora, Sclerospora. Generally, Pythium contains paragenous type of anthracium, but Phytophthora is highly famous for amphigenous type of anthracium. So paragenous type of anthracium is found in Pythium, and also one more special feature is the formation of a vesicle above the sporangium is a special feature of Pythium. So in the question paper number 18, model question paper number 18, we have described about this. This Pythium produces some vesicle or globose like structure that is known as a vesicle. Actually, zoospore formation takes place in this vesicle only above the sporangium. But when it comes to Phytophthora, amphigenous type of anthracium is a special feature. Amphigenous type of anthracium means female reproductive organ, that is oogonium, passes through the anthracium and form a globose structure. Imagine this is the anthracium. This female reproductive organ passes through the anthracium and form a globose structure above the anthracium is known as amphigenous type of anthracium. Such kind of anthracium can be observed in Phytophthora. The next one is the first antibiotic used against plant disease management. Penicillin, areofungin, mancozeb. Mancozeb is not a fungicide. Sorry, not a antibiotic. Areofungin, antifungal antibiotic. Penicillin, world's first discovered antibiotic. Streptomycin, world's first broad spectrum antibiotic. Here, the streptomycin is the antibiotic which for plant disease management started using. So, streptomycin is the first antibiotic that is used for plant disease management. So the answer is the streptomycin. Fruiting body formed in red rot of sugarcane. We must know what is the red rot of sugarcane is caused by Coltrotrichum falcatum. So Coltrotrichum producing asexual fruiting body. We come here. Pycnidium produced by Macrophomina fasciolina diplodia. Sporadicium, Fusarium tubercularia. Acervulae. So Coltrotrichum and Pestalozzi. Cinema, Ceratocystis and Graphi. So here Coltrotrichum is there, right? So Coltrotrichum is produced by Acervulae, right? All Coltrotrichum produces Acervulae are what? Yes, all Coltrotrichum produces Acervulae only. Here red rot of sugarcane is caused by Coltrotrichum falcatum, so it comes under Coltrotrichum Acervulae. Pycnidium is microform and all the other things I have given in this table. So the word bacteriophage was coined by Twart, Herrelli, Slope, Safferman. Twart and Herrelli was individually discovered the bacteriophage in 1915 and 1917 respectively, but the word bacteriophage was the term bacteriophage was given by Herrelli only 1917. Slope, Dello Vibrio 1962. Safferman and Morris probably cyanophages. Safferman and Morris 1963. So the answer is Herrelli 1917. Next one is the reverse transcription mechanism is found in bacteria, virus, viroid, protozoa. So this generally reverse transcription was actually terminology that related to virus. Virus only can reverse transcribe and it can do all kind of stuff. So the reverse transcription mechanism can be observed in virus. Identify which one of the which one caused by the plant pathogen. Black heart of potato, rice caira disease, clubroot of cabbage, a black tip of mango. This black heart of potato is generally caused by two kinds of things. One is sulfur toxicity. No. This black heart. Sorry. Just let me come again. Black heart of potato is the oxygen deficiency, not sulfur toxicity. This black tip of mango is due to boron deficiency and sulfur toxicity, but this black heart of potato is definitely oxygen deficiency. Rice caira zinc deficiency, and all are dengue deficiencies deficiency symptoms except this clubroot of cabbage, which is caused by pathogen Plasmodioporo brassicae. The next one. The conidial cycle of apple scab is completed within. So the conidial cycle of generally the apple scab conidial cycle completed within 8 to 10 days in infected, that is fallen leaves. This is actually happen. Then what is the asexual thick-walled resting spore? They are asking asexual thick-walled resting spore. Whose spore is a sexual spore? Zygospore is a sexual spore. Basidospore is a sexual spore. All are sexual spore. What they asked is the asexual. So the answer goes to chlamydospore. Chlamydospore is the asexual thick-walled resting spore. Okay. But when it comes to sexual resting spore, oospore and zygospores are the sexual resting spores. Parasexuality was reported in Aspergillus niger, Aspergillus flavus, Tamari, nidulans. Parasexuality was phenomenon was discovered by Ponte-Carvo and Roper in 1952 in the organism Aspergillus nidulans. So the answer is Aspergillus nidulans. Fungicide which can move with both the direction upward and downward. Apoplast, Simplast, systemic, amphimobile. Generally, systemic fungicide moves in both directions, but the terminology that is that need to be goes to amphimobile. So the answer is amphimobile. So the next one is clubroot pathogen. Clubroot pathogen severity can be reduced by the application of. So the severity of clubroot can be managed by the application of lime. So 2.5 tons of lime per hectare can be recommended. Generally, this clubroot of pathogen life cycle was discovered by Voronin in 1877. So he recommended that this clubroot of cabbage is severe at acidic soil. So at the pH of 5.2, around 5.2, 5.3 at the time, I mean around 5.2 to 5.5. So if we increase the soil pH by application of lime, because if I apply lime, soil pH can be increased, right? So if we want to increase the soil pH, application of lime is the best management practices at the rate of 2.5 tons per hectare. So the answer is the lime. Application of lime actually can increase the soil pH, so we can reduce the disease. Lichen is an association between lichens are association between a fungi and algae. So algal fungal association is known as lichens. They are very sensitive to pollution. These lichens are very sensitive to pollution. The next one is sex hormone woogonial includes development of woogonium, development of antheridium, meiosis, production of zygospores. Generally, it activates the development of antheridium, so that makes antheridium to develop. So it actually induces the development of antheridium. Antheridium is a male reproductive organ. The male sexual that is in fungi, antheridium is known as the male parts. Oogonium is known as female. Oogonium is produced in Oomycota group of fungi. Ascogonium is produced in Ascomycota group of fungi. Blight cast is the forecasting model used for rice blast, wheat stem rust, apple scab, lead blight of potato. So if we come to rice blast, there is a model called blight cast. For apple scab, mills rules and other things are there, and wheat stem rust also there are plenty are there. But when it comes to lead blight of potato, for lead blight of potato, it is recommended as I mean blight cast is that is actually developed for a lead blight of potato. Gelatin plate technique was given by. So gelatin plate technique was given by Robert Koch. Not only gelatin plate technique, many other techniques was given by this Robert Koch only, pour plate method, spread plate method, such kind of things. That was given by Robert Koch only, and this gelatin plate technique is also given by Robert Koch only. That's why this Robert Koch is known as the father of microbial techniques. Next one is the parasites of the helper viruses. Satellite RNA, satellite DNA, RNA, DNA. So actually this satellite RNA, they required another helper virus. Then they are the smallest. Actually these satellite viruses are the parasites for helper viruses. So the smallest organism capable of independent multiplication, fungi, bacteria, virus and phytoplasma. So phytoplasma can multiply independently, and this is the smallest also. And organism known to induce the production of inclusion bodies, fungi, bacteria, virus and viride. So the production of inclusion bodies are the specific feature or character of plant viruses. So these inclusion bodies also serves as identification factors or determination factors. Plant quarantine is not effective against soil-borne diseases, airborne diseases, seed-borne, waterborne. So this quarantine is not effective against this airborne diseases. The quarantine is especially meant for seed-borne related diseases. The electron dense strain, which is used to determine the internal structures and arrangement of virus particles, phosphotungstic acid, urcyl format, gold, both A and B. So the answer is both A and B. This is phosphotungstic acid and urcyl format. So both can be used to determine the internal structures of virus particles. Long distance movement of virus can be achieved through vascular bundles, plasmodesmata, both A and B, none of the above. So for long distance movement, generally virus goes through vascular bundles only. So if you see plasmodesmata from one cell to another cell, the movement of plant virus from cell to cell movement of plant virus is mediated through plasmodesmata. Generally this plasmodesmata is smaller than that of plant viruses, but still it can manage to go through the tube, that is vascular bundles, with the help of a movement protein. Even though this plasmodesmata is smaller than plant viruses, it can produce a protein called movement protein. With the help of movement protein, it will go from cell to cell. That is actually the easy connection and it can spread very fast. And so the answer is long movement is vascular bundles. The disease which is prevalent only in North India: kernel bunt of wheat, kernel smut of rice, sugarcane smut, red rot of sugarcane. Actually, this kernel bunt of wheat is actually a little bit severe in North India only. This disease was not prevalent, that much prevalent in South India because of environment. Fan leaf effect or shoe sting symptom. Uh, Symptoms is the characteristic features of cucumber mosaic virus, turnip mosaic virus, tobacco mosaic virus, potato leaf roll virus. It is rolling turnip mosaic virus, not tobacco mosaic virus. Alternate green and yellow patches. Cucumber mosaic virus, it produces a short hole symptom or short string symptom or fan leaf effect symptom can be observed in cucumber mosaic virus. The first A virulence gene was isolated from Phytophthora infestans, Fusarium solani, Pseudomonas. Among the given options, the first A virulent gene was isolated from Pseudomonas syringae, Pathology syringae, which causes soybean bacterial something. Pseudomonas syringae, Pathology syringae. The next one is the DNA used an expression vector in the following plant viruses: cauliflower mosaic virus, tobacco mosaic virus, cucumber mosaic virus. Actually, two kinds of DNA expressor will be present in this cauliflower mosaic virus: 35S and 17S or 19S probably. This among the 35S and 19S, 35S is the most well-known and most efficient expression vector, that is we can use from CAMV, that is cauliflower mosaic virus. So the answer is cauliflower mosaic virus. So if they ask for the expressor name, that is 35S. Pathogenicity of agrobacterium depends on RI plasmid, TA plasmid, tDNA, all the above. So generally the pathogenicity is actually dependent on this TA plasmid. TA means a tumor inducing plasmid. This all agrobacterium, the main symptom is production of tumors, right? So that's why it's a TA plasmid, which is agrobacterium tumefaciens, mainly dependent on TA plasmid, that is tumor inducing plasmid. Xylella fastidiosa is responsible for the following: fierce disease of grapevine, phony peach leaf, almond leaf scorch, all the above. So the answer is all the above. Xylella fastidiosa causes, on the right side I have given something: Pierce disease of grapevine, citrus variegated chlorosis, almond leaf scorch, phony peach disease, coffee leaf scorch, alpha dwarf, olive quick decline, all are caused by Xylella fastidiosa only. It is a xylem limited gram negative bacteria. I repeat, it is also the first plant pathogenic bacteria to be completely genome sequenced. If they asked first bacteria to be completely genome sequenced, Haemophilus influenza. First plant pathogenic means Xylella fastidiosa. So Xylella fastidiosa causes all this disease: Pierce disease of grapevine, citrus variegated chlorosis, almond leaf scorch, phony peach disease, coffee scorch, alpha dwarf, olive quick decline, all are caused by this Xylella fastidiosa only. Next generation fungicide introduced in the year, the advanced strobilurin group of fungicide, that is the next generation fungicide, was introduced in the year 1994. Which one of the sexual reproduction method in bacteria requires a cell to cell contact? So generally conjugation is cell to cell contact, transformation no cell contact is involved, transduction involvement of bacteriophages. So the answer for the question given is conjugation. So in conjugation, only one bacteria, donor and recipient bacterium, come in contact with each other with the help of a small tube is known as conjugation tube. We can also call it as X pilus or conjugation pilus. The next one is technique used for the inoculation of a bacterial pathogen. Pinprick and needle method, root dip method, swab method, all the above. So generally for bacteria we can recommend pinprick and needle method. Next one is ring rot disease of coconut and African oil palms are caused by ring rot fungi, bacteria, virus and nematodes. Generally the ring rot of coconuts and African oil palms, probably nematode, I'm not sure, maybe it is caused by nematodes. Potato wart is caused by Olphidium brassicae, Plasmodium brassicae, Phytoderma geometric, Synchytrum endobioticum. So this Synchytrum endobioticum causes potato wart. One more thing, Plasmodium brassicae, clubroot of cabbage. This clubroot of cabbage and potato wart, both the life cycles was discovered by Oronin, MS Oronin. He is a student of Anton de Barry. Most destructive phase of the bacterial disease of rice is known as necrosis, dieback, blight, crescec. So in a bacterial leaf blight of rice, which is caused by Xanthomonas varese, Pathovar varese, crescec phase is the most dangerous and destructive phase. Crescec phase is also known as wilt phase. Next one is viruses which kill bacteria is known as mycophages, phycophages, bacteriophages, all the above. Bacteriophages means bacterial eaters. A virus that infect bacteria or virus that infect and kill the bacteria is known as a bacteriophages. So the answer is bacteriophages. So bacteriophages was first discovered by Twart and Herrelli in 1915 and 1917 respectively. The term bacteriophage was given by Herrelli 1917. Next one is secondary spread of powdery mildew is generally occurred by uh, chlamydospores, conidia, ascospores, cleistocys. The secondary spread is generally occurred by conidia. Which of the following rust is the earliest to appear in a wheat in India? So the yellow rust. Yellow rust is actually first appears in India and comes to Punjab and the north west, where the first wheat cultivation starts. At the time the temperature is very less because yellow rust requires the lowest temperature among all the rust for the infection, around 8 to 12 I think. So at the time the temperature is very less, and the area where especially in Punjab and some other parts of north west, so this yellow rust appears first. So in India the first appearing rust is yellow rust. Fungi predaceous on nematodes. Penicillium, Aspergillus, Glyocladium. Penicillium is general normal mold fungi. Aspergillus also normal molds. Glyocladium is supposed to be a biocontrol. Dactylaria species. Actually, this Dactylaria species is a fungi that actually predaceous on nematodes. So they produce some kind of trappings where this nematode came and traps inside. The next one is antibiotic resistance gene in bacteria is called as S factor, R factor, F factor, M factor. This antibiotic resistance gene in bacteria is known as generally known as F factor. Which wheat rust spreads from Nilgiri and Palni hills to North India? Generally black rust or brown rust, that is a black stem rust, generally spread from this, you believe it to be spread from Nilgiri to Palni hill, Nilgiri and Palni hills to North India. So this inoculum is believed to be coming from Nilgiri and Palni hills. So the first plant virus seen under a microscope: cucumber mosaic virus, tobacco mosaic virus, cauliflower mosaic virus, tobacco rattle-virus. The first plant virus seen under electron microscope is TMV, that is tobacco mosaic virus, that was seen by Krause et al. Krause et al. In 1939. So he was the one who first saw this tobacco mosaic virus under electron microscope. The chemical produced by the pathogen which suppresses the host defense responses is called as suppressors, elicitors, lectins, antigens. Generally, the host producing the host generally contains elicitors. So the pathogen contains receptors. Sorry, the pathogen produces elicitors that was recognized by the host, that is a plant receptors. So to suppress the receptors, the pathogen produces suppressors. So the chemical that is produced by the pathogen to suppress the host defense is suppressors. Which of the following fungi is recognized as a corruption of wheat? Rust fungi, smut fungi, mildew, blights. Generally, smut fungi is known as corruption of wheat. Fungicides in plant disease control. The book was written by Y.L. Nine, Butler, Horsfall, Brown. So this Y.L. Nine, he wrote the book Fungicides in Plant Disease Control. Now the fourth edition was running up, I think. I'm not sure. Fungicides in Plant Disease Control. Now the fourth edition was running. That is a very important one. And D.J. Butler wrote a book called Fungi and Diseases in Plants. Fungi and Plant Disease was written by B.B. Munkur again. The next one, the reserve food material of fungi: glycogen, starch, lipid, glucose. In fungi, the reserve food material is stored in the form of glycogen. For us, however, we have adipose tissue. The fungi has the reserve food material stored in the form of glycogen. Gram-negative bacteria comes under gracilicules, firmicutes, tenericutes, all the above. This gram-negative bacteria comes under gracilicules. Gram-positive bacteria, firmicutes. Both gram-positive and gram-negative bacteria is known as molecules. Response of the host, this phytoplasma and spiroplasma comes under these molecules. Response of the host to the pathogen infection is known as sign, symptom, pathogenesis, allergy. So the response of the host, which means it is producing expressions, right? So these expressions are actually symptoms. So lysogenic strains are those strains of bacteria which involved in virulent phase, temperate phase, both A and B, none of the above. They asked a lysogenic phase, right? Virulent phase is known as a lytic phase. Temperate phase is also known as a lysogenic phase. So here the lysogenic strains are actually related to temperate phases, because the temperate phase is also known as the lysogenic phase. Virulent phase is also known as the lytic phase. Bacterium used in horizontal gene transfer: Erwinia, agrobacterium, rhizobium, pseudomonas. Generally, this agrobacterium used in horizontal gene transfer. Technique used in breeding disease resistance through genetic engineering includes protoplasmic fusion, recurrent selection, embryonic fusion, all the above. This technique of breeding resistance through genetic engineering. So generally, protoplasmic fusion is more related to this question options given, and other also can be used, but compared to this protoplasmic fusion. Heterocyclic which of the following is a heterocyclic compound? Dithiocarbamates, captan, spergon, all the above. So generally captan comes under this heterocyclic group of compounds. To have an understanding about this fungal classification, on the next slide I have given something, I'm sure, I think there we will discuss. Mechanical transmission of a virus is possible that infect epidermal cells, phloem cells, xylem cells, all the above. So mechanical transmission of virus is only possible when it infects epidermal cells. The mechanism involved in disease suppression of Pseudomonas species, SRF production, antibiotic, injury systemic, all the above. So it's supposed to be all the above because it produces all kinds of things. Antibiotics, again it can help in activating induced systemic resistance, and the SRF also it will produce. So answer is all the above. Hostoria is not produced by Pyricularia, Helminthosporium, all of this Pythium. So Pythium is known to not produce Hostoria and Uppersoria. This Pythium cannot produce Hostoria and Uppersoria. Pectate lyases is the major pectolytic enzymes involved in soft rod pathogen species of Pectobacterium, Pseudomonas, Xanthomonas. So they have mentioned something soft rod, right? So which of the following genera is a soft rod genera? It is actually a Pectobacterium. Pectobacterium generally causes soft rod diseases. So the answer is Pectobacterium. This is the classification of fungicide. Inorganic means a sulfur, two types are there, inorganic and organic. Powder sulfur, wettable sulfur, lime sulfur comes under inorganic. Organic means ferbum, zirum, thirum, zinab, menab, vapamil comes under organic. Copper fungicides are like bodo mixture, burgundy mixture, cuprous oxide, chestnut compounds are comes under this copper. Mercury again divided into two, inorganic, organic. Mercury chloride and mercurous chloride are comes under inorganic. Phenyl mercury acetate and methoxyethyl mercury acetate are comes under organic. Phenol fungicide are chloranil and dichloran. Heterocyclic nitrogen compounds. The previous question is heterocyclic nitrogen compounds, all right. Captan and folfate are comes under heterocyclic nitrogen compounds, right. Oxathin, carboxyn and oxycarboxyn. These two are the world's first discovered systemic fungicide by Van Schembling and Marshall Kulka in 1966. This is also important. Oxathin group of fungicide was first discovered by Van Schembling and Marshall Kulka in 1966. The first discovered oxathin group of fungicide is carboxyn and oxycarboxyn. The trade name of carboxyn is Vitavax, oxycarboxyn is Plantvax. Benzimidazole, binomial and carbendazim. Others are PCNB, duodentin compounds and antibiotics are comes under actually. Who is known as the reconstructor of mycology? Anton de Barry, L.R. Tulsen, A. Fisher, J. Kuhn. So J.G. Kuhn is generally considered as a microscopic manager, and he wrote the first textbook in plant pathology that is completely devoted to plant diseases of cultivated crops and their causes and their control, 1858. Anton de Barry, we know, father of plant pathology. And this L.R. Tulsen, generally Tulsen brothers from England, generally considered as the reconstructor of mycology. Actually, the identifications or the discoveries that is related to mycology are like thrown things. So they put all together and make all the things in a proper way. That's why they are known as a reconstructor of mycology. They reconstructed the entire mycology in a proper way. Identify the one with a boron deficiency. Black tip of mango, which is caused by boron deficiency as well as sulfur toxicity. Pahala blight of sugarcane, probably manganese deficiency. Whiptail of cauliflower. Whiptail of cauliflower is due to molybdenum deficiency. Yes, molybdenum deficiency. Rice Keira, zinc deficiency. So among the given options, black tip of mango, which is caused by boron deficiency. Present plant production advisor. So the thing is P.D. Sharma, Rajesh Malik, Ravi Prakash, Trilochan Mohapatra. So while I was writing the book, at the time the plant production advisor is Ravi Prakash. So the present plant production advisor is J.P. Singh. So the present plant production advisor is J.P. Singh is the present plant production advisor. This Trilochan Rajesh Malik is plant production advisor before Ravi Prakash. So Trilochan Mohapatra is the ICAR Director General, ex-ICAR Director General, but the present ICAR Director General is Himanshu Pathak. He is the present ICAR Director General, DG DARE. So the question is the present plant production advisor is J.P. Singh. Okay. So these are all the answers related to the questions given. So further information and furthermore information and preparation of various other kinds of things, this book will be very useful. A Vision to Plant Pathology, complete student version. For further guidance and clarification, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.